So I kind of serve also as the announcer for Gosha's talk that will follow mine. Gosha will talk about the fabulous, and she will explain exactly why we call it fabulous, opportunity that we have been given by Alzheimer Research UK to do research in a new modality in imaging. Well, it's a new and old modality. It's MRI at very, very low field that these days and age can produce decent images. It has the advantage of being cheap and flexible in design and can be dispatched in, for example, memory clinics across Sussex. So there is a good incentive to look at the possibility of using this type of scanners to see whether the quality of the images are good enough to provide the same imaging data that is used as a corollary for diagnosis that we get from the more expensive and fixed machines in big medical centers. But I will leave all of that to Gosha to talk about and proceed to my topic, which is to convince you that there is MRI beyond structural MRI and that there is MRI that is relevant to dementia beyond those structural measures that I'm sure many of you are familiar with. So this is just an example of how structural MRI is used in, for example, classifying different types of dementia, or at least hinting at the possibility of classification, and also grading white matter damage, which scales probably with vascular deficits. So all of this is very nice. Question is, A, how useful it is, and B, where is it positioned with respect to what we call the set of biomarkers that tell us about not only the existence of dementia, but also about its severity. And you can see that structural MRI falls, kind of in a mediocre place. I'll turn on a marker here to have a laser pointer. So we are roughly here. So yes, before changes in cognition become prominent, but way after many of the physiological, neurochemical, and functional changes that are characteristic to the evolution of dementia happen, and those changes are detectable with other methods. And the question is, for example, if MRI can provide an alternative method to look at metabolic patterns or changes in metabolic patterns in the AD brain in the way that, for example, FDG PET does. Or can MRI, for example, tell us about the presence of aberrant proteins, and in particular in intracellular proteins like tau neurofibrillary tangles in the AD brain, either to map it or to measure it localized in regions where we know that are part of, for example, of the BRCA progression, disease progression, and we would like to see whether there is a reliable measure that correlates with tau aggregation in those regions. Similarly, we would like to see whether there is a hint from MR measurements regarding the response of the innate immune system, and in particular in microglial activation, but also in astrocytic activation. And you can see, for example, here that based on post-mortem GFAP studies, astrocyte dysfunction is actually one of the earliest events in the evolution of Alzheimer's disease. So if there is something that can be measured with MR that reflects the same type of dysfunction, that would be a big bonus. The first thing that I want to try to talk about is whether we have a proxy to FDG PET or a proxy to metabolic measurements using MRI. And I will try to convince you that we do have. And this particular methodology that we do have is related to measurement of perfusion or cerebral blood flow, but not in major blood vessels, but in the capillary bed, where the major event of exchanging with the parenchymal tissue is taking place. And for that, I will run you a crash course in what we call arterial spin labeling, which is the main method in which we measure perfusion. And the idea is relatively simple. When we measure with MR in general, we're looking at a signal that is generated by what we call spins, but those spins are essentially the nuclei of hydrogen atoms. And since the carrier of hydrogen atoms, which is found in the largest abundance in the body, is water, essentially MRI reflects in one way or the other the amount of water dispersed in tissue. So if we do a measurement, this is what we get. But we can also, what we call, label the spins that enter the brain through the major arteries that feed the brain and invert those spins. And then the image that we get will be a mixture of the signal of the innate spins in the brain and those that freshly arrived from the labeling plane around the neck. And we perform this measurement alternately. And eventually we end up with two types of images, one with a label and one with control. We subtract one from another. And if you have kind of a very kind of sharp sight, you can see that there is a faint image of something that looks like a brain. But that's not enough. So you have to average this for about 5 minutes of acquisition. But then you do get something that really looks a little bit clearer. Not only that, but you can really see that this type of image gets very strong signal from gray matter, from cortical gray matter in this particular slice, and weak signal from white matter. And this is because the amount of label that came from the neck arrived more to gray matter, because gray matter is more heavily perfused than white matter. And then by miracles and mathematical operations, one can translate this image to a quantitative map that describes cerebral blood flow. So this is all good news. And the good news become even more spectacular when you look at paper like this from our good friends at UPenn. John Detry is one of the founding fathers of the field of arterial spin labeling to measure cerebral blood flow. Just to give you an idea, the maps that you see here, so the lowest row is an 18F FDG PET map, and the maps above are CBF map using ASL. And they're all scaled, of course, to look similar in terms of the color range, but also the patterns that you see are very similar. And these are done in controls and in MCI patients. And you can see, for example, that in this case, where you would expect to see the posterior cingulate lighting up, you actually see it also lighting up in the difference map or in the perfusion map that comes out of arterial spin labeling experiments using MRI. So this is a good evidence to the well, not I'd say equivalence, but parallel types of information that comes from metabolic measurements and vascular measurements, all made possible through what we call neurovascular coupling. So things that are happening on the neural level have an interaction with what is happening on the vascular level on a spatiotemporal domain. So brain metabolism is linked to brain perfusion, and this allows us to indeed indirectly measure metabolism by looking at perfusion. This is a study that was performed more than 10 years earlier than this study by a member of our team, Iris Aslani, who at that time was in the imaging center in Columbia University in New York. And they already reported ASL quantitative assessments of ASL measurements in healthy controls and in Alzheimer's disease patients, both left and right hemisphere, in a long list of different brain regions. So all that looks promising, but then it is important really to know what you're doing. And I just mentioned this because the same Iris that now works in our center also developed a method that I will briefly describe here to correct from one of the most common hurdles to proper interpretation of ASL data. This is a study that looks at perfusion differences between healthy controls, SC versus AD, healthy controls versus mild cognitive impairment patients, and so on. And you see that when you subtract maps from two subject populations, you get the difference map that tells you where is the deficit in perfusion that you would expect to see in this particular population, in this case, AD or MCI and so forth. And when you look at this map, it looks very, very extensive. Again, same regions are the suspected regions. You see here the PCC. You see here, for example, the temporal lobes and so on. However, one has to remember that we're looking at a relatively low resolution method. And in low resolution method, we have problems of what we call partial volume effect. In the same unit of image, which we call voxel, there are contributions from both gray matter, white matter, and also CSF. And if the cortex shrinks with age and with disease, then the tissue fraction of each type of tissue within your volume changes, and you don't know whether changes in perfusion are caused by physiological effect or by shrinkage of tissue. So Iris, together with her colleagues in Columbia University, developed a method to assess and to correct for this partial volume effect. And when you correct for partial volume effect, then you see that the effects are A, more localized, and B, more meaningful. And this is not, by the way, a study that just implemented her method to measure perfusion in AD. And the interesting result that could have been a positive result, for example, in this case, blood flow decreased in the hippocampus in patients with AD compared with subjects with subjective complaints, the effect disappeared after partial volume correction. So it's important to know what you're doing and to implement all the entire battery of tools that you have in order to get reliable results. So that is my spiel about physiological MR, and now I will tell you a little bit about neurochemical MR. And neurochemical MR is a modality that we call magnetic resonance spectroscopy. And if we hark back to what I mentioned earlier, that the signal that we detect in MRI comes from the nuclei of the most abundant proton or hydrogen atom carrier, which is water. Once you get rid of that signal, you have the contribution of hydrogen atoms that are not water, that are within our detection limit. And detection limit typically limits the concentration of the molecules that we can generate signal for or from to the roughly millimolar range and above. And within this range, there are several molecules that are detectable with magnetic resonance spectroscopy. By the way, as you can see here, the detection of magnetic resonance spectroscopy is done typically with selecting a box from which you extract your signal. There is also a possibility to get it in a kind of a coarse map across the brain. There are pros and cons to those different types of acquisition. I will focus on what we call localized spectroscopy, so spectroscopy from a box. And within this box, you get signal that comes from metabolites that some of them are purely or almost purely neuronal metabolites, the most famous of which gives rise to the strongest signal that we get in spectroscopy, is a mysterious molecule called N-acetylaspartate. N-acetylaspartate is possibly synthesized within mitochondria, but it is prevalent mostly in the cytoplasm of central nervous system neurons, and we accept it to be a marker for neuronal viability. Of course, the number of neurons will also determine how much N-acetylaspartate we have, but also how functional those neurons are. The other one is glutamate, which is the most abundant excitatory neurotransmitter, and also GABA, which is less detectable, and it is the main inhibitory neurotransmitter. But there are also metabolites that are found predominantly in glia. Glia meaning astrocytes, microglia, and oligodendrocytes. But in the case of this molecule here, myoinositol, we're talking mostly about the astrocytic population. Same thing also about different types of choline compounds. Here is the choline compounds, and here is the myoinositol that you can detect and quantify, and will tell you about, for example, astrocytic activation or astrocytic concentration in the case that we have astrocytosis and increase in the number of glial cells, including astrocytes. And there are metabolites that are equally distributed across cells. famously the pair creatine and phosphocreatine involved in cellular metabolism. Relevance to dementia has been researched quite extensively across different groups in the world. One of the most active ones is the group of Cliff Jack in the Mayo Clinic in Minnesota, and Kajal Kantarchi already in 2013 did spectroscopic studies in the posterior cingulate showing that with the increase of pathology, you have an increase of myoinositol, meaning some sort of effect related to probably astrocytes, and decrease in NAA, meaning decrease in viability from, again, healthy controls all the way through MCI to MD. And we had an interesting collaboration with a group in Lund University. Oskar Hansson in Lund is a neurologist that leads one of the largest studies in AD research in Sweden. This is the Biofinder study. We were lucky enough to latch onto this study and do a couple of studies related to it. One looked specifically at concentration of myoinositol in conjunction with both A-beta distribution and tau distribution as measured with PET ligands, both for tau and A-beta. And just to give you, for example, an interesting result that we obtained is that there is an interesting mediation of beta and myoinositol via ApoE4. So we see here, for example, that if we look at the concentration of myoinositol as a function of beta signal as measured with amyloid beta PET within the posterior cingulate, overall, over the general population, there is no correlation. But for ApoE4 carriers, there is a strong correlation between the concentration of myoinositol, again, astrocytic activation or proliferation, with the concentration of A-beta within the volume in which we performed the measurement. And I want to conclude before handing the baton to Gosha with a relatively rarefied atmosphere, sophisticated measurement that uses spectroscopic signals not to look at their concentration, but to look at their mobility. And why is their mobility so pertinent to disease? Well, this is something that has been discovered maybe about 50 years ago, when people noticed that there is an interesting change in the diffusion properties of water in disease, most famously in stroke, but also in a host of other diseases, including neurodegenerative neurological diseases of other types, like, for example, in MS, changes that relate, for example, to demyelination. If we look, for example, at changes that we expect to see in cytomorphology related to Alzheimer's disease, then we have the extracellular accumulation of amyloid beta, but the intracellular aggregation of abnormal tau in neurofibrillary tangles. This is from the neuronal side. From the glial side, activated astrocytes and activated microglia change their shape or their morphology when they are activated. And that means that we have a handle to look at those changes if we have the ability to measure the mobility or the diffusion properties of the metabolites that we said that are specific to neurons or to glia. So this is just to give you a pictorial example. If we go from a healthy neuron to a neuron that is laden with neurofibrillary tangles, with tau neurofibrillary tangles, then we would expect that the intraneuronal diffusion will change, either become more facilitated or less facilitated, but let's assume that those kind of aggregates create a barrier to diffusion. Then we would observe a decrease in intracellular diffusion of all the metabolites that reside specifically in neurons. Conversely, glial activation changes the morphology in such a way that we would expect to see changes in the intracellular diffusion of metabolites that are expected to reside in astrocytes and microglia, in particular, the famous inositol or myoinositol that I mentioned earlier, but also in choline compounds. This gave us the impetus to, again, latch onto the Biofinder study and to conduct a study in which we don't measure, well, we measure in addition to the concentration of metabolites, also the mobility or the diffusion of metabolites in the posterior cingulate. And that study, by the way, also looked at the concentration of a tau PET ligand that they administer to the patients. So we have both PET data, but also spectroscopic data and diffusion spectroscopic data from this particular region in two cohort groups. So we have Alzheimer's disease patients and healthy controls. And this is just to summarize what we found. This is a slide that shows you the changes or the differences in concentrations between cognitively impaired, or AD, in brown, and cognitively unimpaired, which are healthy controls, in blue. And these are the concentrations of metabolites, essentially replicating what Kajal-Kantarci and Cliff Jack found in their cohort. We have a decrease in neuronal viability shown by decrease in NAA concentration, increase in glial markers, choline compounds, and myoinositol. There is an interesting relationship between the diffusivity of water and the concentration of myoinositol, which gives us kind of provokes us to think that maybe the diffusion of water, which I don't really want to talk too much about in this context, might reflect also passage of water through aquaporin channels in active astrocytes. But the main event here is our finding with respect to the diffusivity of metabolites, and in particular, the changes in diffusivity of neuronal markers, going from healthy controls to neurons that are affected by neurofibrillary tangles, from healthy controls to AD. We see a fascinating or almost unique change that you don't almost see in diseases, which is decrease in mobility of this intraneuronal molecule, which we hypothesize that is related to the aggregation of tau within neurofibrillary tangles affected neurons. Just to show you that this is a unique trend, when you look at the diffusivity of water that reflects diffusion in all tissue compartments, including the extracellular space and astrocytes and glia and oligodendrocytes, we have a net increase in diffusivity. So this tells you that we are measuring something that is more specific and possibly related to this particular intraneuronal pathology. So the diffusivity of NAA is specifically modulated by intraneuronal pathology, whereas the diffusivity of water is modulated by the sum of all microstructural alterations in AD, thus less specifically tells us about what is happening within neurons or within glia for that matter. So the take-home message is quite simple. MRI is a multimodal measurement. Structure is just one of many dementia-related metrics that MRI can provide. Perfusion tightly correlated with energetics, and it should be, I think, more deeply explored as an early marker for different types of dementia. And the spectroscopy provides a completely different angle into cell-specific pathology, both via concentrations, but also via other properties that, let's say, with more advanced methods, tell you about specific pathologies pertinent, for example, to Alzheimer's disease, but also to other types of pathology in primary tauopathies, which is something that we plan to explore with other collaborators in this country and abroad. So, well, there is more, but I haven't talked about more. We focused on two modalities that are non-structural, but there is more to be aware of. For example, MRI that is sensitive to iron aggregation, MRI that is sensitive to permeability of the blood-brain barrier, and so on and so forth. So I think I did my role as a used car salesman for the multimodality of MRI, and I thank you very much for your attention so far.